Hello, everyone, and welcome to today's webinar, High Throughput, High Resolution Purity, and Heterogeneity Characterization of Biologics using Breakthrough Multi-Capillary Technology. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SCIEX. To learn more, visit sciex.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Fang Wang, Technical Product Manager at SCIEX. Dr. Wang, you may now begin your presentation.
Thank you for the introduction and thank you all for joining us today. My name is Fang Wang. As introduced, I was an analytical scientist working in the biological characterization in the biopharma before I joined SCIEX. I still remember that saying, right? I wish we can just analyze them all. And all the other conversations in that sample planning meeting. How many samples can we submit? Did we submit enough? And are the samples we submit representative? And also working in the lab, I have to calculate the number of reagent wells to build a sequence and calculate the start time for each step to create a method. Sometimes I have all my reagent and samples ready and walk to the instrument only to realize the instrument I signed up for doesn't have the right detection module. Or when a set of samples come in and they are critical to be done and I'm at home, but keep wondering, how is that P100+ doing overnight? Or the time I wish the data processing can be more robust and simpler. What if there is a system that elevated these issues, took away these pain points from the current P100+ system and allowed you to develop a higher throughput method but keep the same high quality data? With that, I would like to share with you the high throughput, high resolution purity and heterogeneity characterization of biologics using a breakthrough multi-capillary technology. So in recent years, changes in regulatory and market dynamics have also created challenges in biopharma industry, forcing the developer to address factors around time to market for new biologics. Additionally, overcoming challenges like sample handling limitations, increased molecular diversity, time-consuming characterizations, all while simplifying the workflow has become a focus for many in order to enhance their productivity while maintaining the safety profile. The BioPhase 8800 system gives you the agility to analyze up to 8 samples simultaneously with automatic capacity of up to 96 samples. And we see this platform as a complete characterization solution in which a multi-capillary array will enable processing of more samples in a shorter time with the same high-quality results you currently have on the existing cIEF, and giving the biological development opportunity to take charge of the biological pipeline and accelerate time to market. The BioPhase 800 system inherited many of the essential features on the existing P100+ platform, like the multi-modes of detection, the sample and capillary temperature control module, the onboard pressure delivery system found in the existing system. However, each of these features has been enhanced to deliver an improved user experience while minimizing the requirement for additional touch points and enables the instrument to incorporate a greater level of automation for operation in a high throughput environment. And the system is strictly designed to maximize the system operation and maintain its robustness. For example, incorporating a software-controlled modular detections eliminates the need for manual switching between detectors as it is now. And it is also incorporated on-board instrument control touchscreen and allows you to recall sequence, perform direct control actions, and do system monitoring right in front of the instrument with this touchscreen. And in the new software, we also provide simplified and more intuitive master and sequence editing capability. It lowers the barrier for operation training and also gives the ability to data integrations and multiplex data processing. So the user experience has improved significantly and allowed more samples to be tested, potentially freeing resources for other activities. The system and software also provide support to establish 21 CFR compliance, giving a QC-friendly setup. So the innovative pre-built all-in-one 8 capillary cartridge eliminate a lot of the needs for manual build cartridge and take care of the interface, and give us a ready-to-use environment where parallel processing of 8 samples can be performed. The liquid temperature control of capillary maximizes the data reproducibility from capillary to capillary and from injection to injection. It also gives the ability to multiplex by batch analysis to speed up potential design of experiment from weeks to days. It shortens the master development and optimization time and qualification times from months to weeks and days. So in addition to the engineered robustness in the system in general, we also put great efforts into streamlining the entire workflow. We took the assay which the current CE users are using and already familiar with, designed and developed them specifically for operation on BioPhase system, that including the assay kits with the colored cap, reagent cap, and application guide for sample prep and system operation. For users currently familiar with the single capillary P100+ system applications, the consistency between the chemistry also gives a great opportunity for master transfer from existing P100+ system without much master development requirement and efforts. When the capability and functionality enhance characterization workflows, BioPhase 800 represents the bridge between the upstream high-throughput setup and downstream sample and processing, thus laying a foundation for consistent analysis through the biopharma development continuum using a single capillary-based platform, and from upstream to downstream with a single platform and give us a great opportunity for successful method transfer too. So BioPhase is not intended to simply operate just as another instrument or a 8 capillary P100+ system. Rather, it incorporates an end-to-end workflow consisting of sample prep, system setup, and all the way to data processing. And we begin by developing a separation method and run sequence using an intuitive tile-based drag and drop interface, where the users can use the software and select desired samples, buffer reagent, and specific actions like wait, rinse, inject, and separate to assemble into a master and a sequence timeline. This can be performed in both remotely and on a workstation that's in the lab. Following the sequence development and upload, a map for both sample trays and reagent trays can be printed out, and users would take that map and populate the trays and BioPhase instrument with appropriate samples and reagents before moving on to the separation itself. On the front panel touchscreen, the user can recall the sequence and initiate the run. Once the separation is underway, progress can be monitored using the touchscreen panel on live. And finally, once the separation is complete, data is batch transferred to the workstation or the server that can be processed using the embedded BioPhase analysis module. And the BioPhase analysis module is with improved robustness and algorithm allowed a very automatic data processing in a batch setup. So much like a P100+ system, the BioPhase 88100 system can support multiple applications for characterization of traditional maps, such as our classic CSDS assay and the identity and characterization with the high-resolution cIEF. It also supports the applications for cell and gene therapies, such as capsid protein purity analysis, empty and full capsid comparison, genome integrity, and mRNA analysis. With that, I will take a snapshot of some of these existing applications and demonstrate the capability. So what we are showing here is a single injections of reduced IgG control standard on the 8 capillary platform. As we can see, between the capillary A through H, we have a high reproducibility in terms of migration time of the marker, the light chain, and the heavy chain. We also demonstrated excellent resolution between the non-glycosylated and glycosylated heavy chain, as we have been seeing on the existing platform. We are also able to demonstrate the linearity and evaluate the detection quantification limit on the platform in both UV detection mode and LIF detection mode. Shown on the top is the linearity demonstration of NIS IgG, and with that data, we were able to demonstrate the LLD of 2.5 microgram per ml and LOQ of 4.9 microgram per ml. And all the 8 calibration data points can be obtained from a single injection, reducing the time for method validation on the linearity part from 8 hours to a single hour. And as mentioned earlier, the switch between the different detection module, UV and LF, is controlled through software. No need to manually switch on the hardware part. The same sequence, the different detection can be performed within the same sequence. Shown on the bottom is a linearity assessment of this CSDS using a pre-labeled NIST map. And in this particular case, we label the NIST IgG using a fluorescent dye and subject to the same CSDS analysis. With the results, we were able to show 3 orders of magnitude reduced in terms of limit of detection, which means a 3 orders of magnitude of sensitivity increase. And the LLD and LOQ was reduced to 4 nanogram per ml and 10 nanogram per ml respectively. Again, all the 8 calibration data points can be obtained from a single analysis. Another important application of the CSDS is to analyze and quantify impurities in the sample. We demonstrated that capability using a spiking assay. In this particular setup, we spiked in 1, 10, and 100 microgram per ml of the lysozyme into a 1,000 microgram per ml IgG control standard. And after the reduction in sample prep, we were able to detect as low as 0.1% lysozyme spiked in with about 90% accuracy and recovery. The signal-to-noise ratio, even with the lowest spiking, was shown to be about 25, way above the limit of the quantification requirement. That demonstrates that the current platform can able to provide detection of impurity as well as 0.1% confidently, which matches the regulatory requirement. And in addition to the CSDS assay, a complete assay kit was developed to force BioPhase 800 system. The capillary isoelectrophoresis and charge heterogeneity analysis can now be performed the same way as the existing P100+ system. And here we are showing a comparison of the NIST IgG charge head data on the P100+ system on the right and on the BioPhase 800 system on the left. As we can see, very comparable resolution can be observed for the different charge variants on the NIST IgG map. And also, we prepared the sample by spiking 4 different PI markers to determine the PI value of the sample. And during the data process, all 4 PI markers was included to generate a linear calibration curve for the determination of mean peak PI values. And in addition to the system comparability between the two platforms, we also performed repeatability assessment of the cIEF kit on the BioPhase system. And showing here is we did a 6 consecutive injection of NIST IgG cIEF samples on all 8 capillaries simultaneously, giving us 48 data points in 6 hours. The profile correlates very well between run to run and between capillary to capillary. What shown here is a single injection overlay of all 8 capillaries, but the percentage RSD calculation included all 48 data points. As we can see, the percentage RSD for reported percentage mean, percentage acid, and percentage basic are all below 2%. The highest value comes from the acidic variants, as that's the most low abundant species in that profile. Demonstrating great reproducibility, and the new assay kit eliminates the need for sample reagent preparation for the cIEF analysis. So in addition to the protein and the analysis that's supported on the platform, we also demonstrated the capsid protein analysis purity assay on the BioPhase 8800 system. And in this, during the transfer of the assay from P100 Plus system to the BioPhase, we were able to perform the parameter optimization for 3 different parameters within 4 hours, taking fully advantage of the multiplex capability on the 8800 system compared to the 48 hours that we did before on the P100 Plus system. So here is a side-by-side comparison of 3 different serotype AAV and for their capsid purity analysis. On the left is the results from P100 Plus system, and on the right is results from the BioPhase 8800 system. So with that, as we can see, the migration time aligned very well between each other, and the profile for different peaks, the number of peaks and relative intensity also consisted between the 2 platforms. As expected, the migration time and the distribution of species aligned very well, and during the mass transfer, there's minimum change between the 2 platforms. In addition to protein capsid analysis performed for the AAV capsid, we also demonstrate capability to analyze AAV genome integrity on BioPhase 8800 system. As we can see here, we used 3 different types of AAV, AAV2, AAV5, and AAV9 with similar genome setup encapsulated, and with that, we were able to analyze the same profile and report the similar percentage of purity between the 2 platforms. The most purified sample in here, and also showing different level impurities. In the sample we obtained, the AAV9 has the highest genome integrity profile, while in AAV5 for the middle and AAV2 for the relatively less purified samples. And we were able to demonstrate the capability to analyze all kinds of purity level for the genome integrity. To summarize, I gave a brief introduction of the BioPhase 8800 system and also showed a snapshot of multiple applications that can be supported by this platform. We have an easy-to-use software with real-time monitoring on the touchscreen, and the system allows switch between LIF and UV detection module controlled by the software without the manual switch. And with the 8 capillary cartridge, one can not only speed up the method development by parallel processing, but also do a near real-time analysis. With this system, we can bridge between upstream and downstream environment. The upstream high throughput can be achieved by the multiplex system, while the downstream is supported by the 21 CFR environment. And the last, thank you for your time and attention. I can open to take any questions.
Thank you, Dr. Wang, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what cartridge options are available? Can you build your own cartridge?
Yes, so with the current system we have commercialized, and the cartridge available is the same as the pre-built 20 centimeter effective length and 30 centimeter total cartridge. And the for the cartridge, BioPhase system is intended to provide you a easy, all-in-one, like ready-to-use system. So the cartridge, essentially you can't change the cartridge by yourself. It's everything included in one device. Once you have the cartridge, you will be able to recognize you essentially we eliminated the interface cleaning, the electrode build, and everything, and put them into this one device. All you have to do is insert a cartridge into the instrument. That of course take away some of the flexibility we had in the P100 class. I hope that answers the question.
Great, thank you. Next question. How can the BioPhase 8800 speed up method development? Any examples?
Because we essentially, the BioPhase 8800 system provided a multiplex platform, allow you to run 8 samples simultaneously. And with that, we were able to, in our hand, we were able to demonstrate the method validation that include LOQ, LOD detections, and the linearity with different concentrations and different voltage for the same sample analysis, different temperature, all that within one sequence. So essentially, before I would say that requires probably 2 or 3 overnight sequence. Right now, you can done in one setup in overnight sequence. And also, we were able to do master optimization, as I shown in the slides, for the AAV genome integrity workflow. And before, we did the same essentially evaluation and development work on the P100 Plus system for our earlier release last year. And for that work, we have to take 72 hours minimum to complete all the experiment. But for here, we were able to do in one sequence.
Thank you. Next question. You mentioned something about building method and sequence remotely during presentation. Would you please elaborate a little more details?
Yeah. So what we are providing is trying to separate in terms of space and workflow what you can do without using the wet lab versus something you have to do in the wet lab, such as sample prep and reagent and all chemical handling. And so the current system is designed to have the method, the sequence build, and data analysis all taken away from that computer or the workstation we used to have right next to the P100 Plus system. So what that also means is we have the server-based configuration, and all the data method sequence goes onto the server as long as you have access to your network, and you can build your method and build your sequence in there. And once the sequence is built, actually, I think a lot of us remembers you have to do the wild calculation and volume, right? And now the report will give you an exact map of what's needed, the volume reagents, the number of wells or not wells, the wells on that 96 well plate to fill, and give you all that information. And you can print it out into a PDF or into a paper and bring that paper to the lab to fill your tray. And once that tray is there, and you can load on the instrument and touch go. So then by that point, or your lab work is done, you can go back to your computer, do the data analysis remotely, and the data is uploaded or transferred after every single injection. So you can monitor the data relative much sooner from a remote setup compared to before.
Thank you. Next question. What applications are available on the BioPhase 8800?
We have validated CSDS and the cIEF, and both of them will come as a kit setup. As I shown, right, the reagent is color-coded with what's used in the software, and that's the two kits validated. But we were able to essentially run a lot of the other applications previously on P100 Plus with minimal modification, as long as the required cartridge or capillary is 20 to 30 centimeter, 20 centimeter effective and 30 centimeter total. And we have both spare fused silica and the neutral options. And for cIEF, we use the neutral cartridge, and for many other applications we published as the technical notes, they were using the bare fused silica version. So yeah, in a lot of things you were able to run on P100 Plus can be tried at least on the BioPhase system, but what we validated as kit is CSDS and cIEF.
Thank you. Next question. Can the new system be used for glycan analysis?
For the glycan analysis, the kit is a follow-on release. So as the effort for BioPhase, right, we're trying to provide a continuous improvement or conditioning, a continuous add-on system for this particular platform. Since new kits and new workflow will be gradually added onto the platform as a validated kit. Right now, it's not yet a validated kit, but again, as I said, we have the 20 30 centimeter cartridge available. So if your particular method is using that cartridge, there's nothing against you to test it on it. I hope that answers the question for, yeah.
Great, thank you. It looks like we have time for one more question. Since the vials caps for the samples were traded for a 96-well plate, are the solution buffer vials also replaced with 96-well plates?
The short answer to that question is yes. So we have essentially the entire configuration changed into a plate-based setup. And for each run or sequence, you need 4 sets, similar like before, like P100 Plus. There's 4 locations. You have the sample inlet and sample outlet, and reagent inlet and reagent outlet. So the outlet tray are designed the same for both the sample and reagent. But for the sample inlet and reagent inlet, we use different form of 96-well plate. And the sample plate, we're trying to reduce the volume required compared to the reagent. So, but all parts is available, and if additional pictures or details is needed, feel free to reach out to us.
Great. Well, thank you again, Dr. Wang, for your time today and your important research. We would also like to thank LabRoots and our sponsor, SCIEX, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.